Okay, this study was kind of born out of an interest in studying human bone marrow tissue using more sophisticated tools and techniques and forms of data analysis than we have been using historically. For those in the audience and those listening, we routinely review human bone marrow tissue for a variety of indications as part of routine clinical diagnostics for both malignant and non-malignant conditions, but many more for malignant states. These include leukemias, lymphomas, myeloproliferative neoplasms, diseases like multiple myeloma, for example. And the goal of the study was basically to demonstrate that we can use those types of tissues and identify many different cell types using antibodies and fluorescence labeling, and then image the tissue using a slide scanning technique, and then use data science with artificial intelligence to identify where all potential cells of interest are located and determine the relationships between them, where they kind of lie next to one another, and whether any of that has biological or potential clinical implication. And this initial study was a spatial mapping of human hematopoiesis using these types of tissues, basically to demonstrate the capability to do this. And I think it lays the foundation for a lot of ongoing and future investigation using this tissue type and a similar technique, but primarily focused in hematologic diseases. And one of the areas of ongoing focus is in myelodysplastic syndromes, where evaluating bone marrow tissue remains the gold standard for analysis. But as I'd mentioned, we're using a lot of kind of historical approaches, and I think we have potential to bring this into the next century with the way we analyze these tissues to help patients and guide therapy. So I think one of the hopefully promising aspects of this particular approach is that we utilized a tissue staining and imaging kind of protocol that's been around for at least about 10 years now. The imaging system is actually based within our CLIA laboratory, not that we use it for clinical application just yet, but we have the potential to move it in that direction by virtue of its location, a lot of the automation that's involved in the technical aspects of the tissue staining and slide capture. And then, as you mentioned, of course, the data science is a tremendous requirement. Without that, we can't really analyze the data and generate the kind of results that we produce and kind of describe in the study. And so I think that's going to become a necessary component of any sort of analysis of this type. And certainly in the clinical space, if this were to move in that direction, data science would remain an important component. But artificial intelligence in healthcare is going to be prevalent in a lot of different areas. AI for medical imaging is a very rapidly evolving area. It's very prominent in radiology already, and I think pathology is probably the next area where it will take hold and help us really move the needle forward. The goal of this initial study was to highlight, hopefully for the community, that in routine clinical practice, we collect this particular type of specimen from patients, and historically it goes relatively underutilized following the initial diagnostic evaluation. So one of the goals of this study was to demonstrate that that sample still exists, and using that and using more sophisticated methods for analyzing it, there's an opportunity to learn a lot more about disease biology, but also to better categorize patients. That definitely may carry with it both therapeutic and management implications.